in near Sedona, Arizona, USA, is actually happen through weathering. Okay, and the previously it is full with rocks, but due to this weathering process, so we can see this hollow alcove within this particular rock. Okay. Now for this one, we have chemical and biological weathering. One. Okay, so the left part here, okay, this left part here, you can see that, okay, biological weathering by lichen. So you can see the vermilion color or yellow and orange color, dark yellow and orange color structure here is actually it is actually a lichen. Okay, lichen is basically a combination of fungus and algae, and if they form symbiosis interaction within them and eventually form into a lichen structure like this. Okay? And this can be seen at La Palma. I think, and this one can be seen also at in Cameron Highlands. If you go to Cameron Highlands, and so you can see this kind of lichen system. Okay, I need to pick up the call first. Okay? So just wait for a while. Okay, I've mentioned about the lava, the lichen. Okay, biological weathering. Okay, of the lava. Lava is actually, this one is a solidified lava. Okay, from the volcano. Okay, and we can see that the lichen start to grow, and eventually, what will happen to this lava? It will start to become brittle and dismantle, and eventually, it will fall to the soil, and this will also add up to the or increase the heavy metals, okay, or the metals concentration on the soil. Okay, and on the right side, this one is actually the chemical weathering. Okay, comparison of the unweathered. Okay, you can see here unweathered is the left part here, and the weathered on the right part. Okay, of this particular picture. Okay, within one set you can see unweathered. Okay, you can see the structure of the unweathered chemical weathering. This one, no action on the chemical weathering, and the right side you can start to see, okay, something is has been corroded here on the surface of this boulder. Okay, this is due to the chemical weathering, and this happened on this particular limestone boulder. Okay, right, and then we have finished with the weathering, okay, chemical weathering and so on and so forth, and now we want to explore the the biosensor part. Okay, biosensor part. Okay, so why this topic is very important, because we actually, okay, from this the interaction, okay, of this microbe and also the metal. Okay, so we can actually use this special interaction of microbe and also metals, okay, to actually develop biosensors, okay, to detect the metals. Okay, okay, so how to do this? Okay, by using the metal specific bacterial sensor in addition to chemical analysis, it is possible for us to determine, okay, the presence of the metal, okay, from one sample to another. Okay, so this is called as biosensor. So recently there are many metal specific sensors have been developed and applied in the laboratory. Okay, in table 1, I'll show the table 1 after this. Okay, on the next slide. Okay, and these sensor strengths are all based on the same concept, which is a metal responsive regulation unit. Okay, that regulates the expression of sensitive reporter gene. Okay, so meaning there is a, or when this particular microbe detect can detect the presence of certain type of metals, okay, in the environment. So it will start to the microbe, once it senses the presence of this metal, it will start to trigger the gene expression unit, okay, within its cells, okay, within the nucleus of the and this will be triggered the DNA basically to conduct the expression of the sensitive reporter gene. Okay, so the reporter gene will be expressed, and this can be tied up with certain bioluminescent proteins, okay, such as bacterial luciferase, LUX-AB, the firefly luciferase or LUC-FF, or can be as simple as for beta galactosidase, okay, which can be detected electrochemically or by using chemiluminescent substrate detection. So what will happen, okay, when the microbe detect the presence of the metal at one site, okay, when this particular reporter gene is being expressed, okay, meaning when I say expression of gene, the S244 and I think S257, you guys will know expression of gene, what will happen is protein will be produced, okay, or maybe enzymes or protein will be produced. So this, for instance, bacterial luciferase, okay, this is an enzyme, okay, that will make the chemiluminescent substance will glow, okay. If you talk about bacterial luciferase, and if you talk about the using the firefly luciferase, we are not using the firefly directly, but we take the gene, okay, from the firefly luciferase, encoded into the bacteria through molecular biological technique, okay, genetic modification, and eventually we can encode this LUC-FF into the cell. And when the cell is expressing, okay, when metal is present, then the cell will the sensitive reporter gene will express the luciferase, and eventually when there is a chemiluminescent substrate within the system, the substrate will glow, okay. So if the substrate glow, we know that, okay, the metal is present. So this is what we call as biosensor, okay. So the light produced can be measured by various instruments, including luminometers, photometers, and liquid scintillation counters, okay, right. Okay, this one table 1, okay, as I did mention to you earlier. So if you want to detect antimony, okay, or SB3+, okay, so the reporter gene is LUC, okay, luciferase, and the host strain we can genetically modify the Coccus aureus, Bacillus subtilis, and so E. coli to get this thing done. Okay? And we have also LUX gene, okay, like as I did mention to you earlier. We also have GFP, green fluorescent protein. We also have LAC gene, okay. And I think in the most used gene is actually the LAC gene, okay. All right. Okay, so now we go to this particular. Okay, this is the operon. Okay, the operon of how we can construct the biosensor. Okay, how to construct biosensor. So the structural genes here. You know that this is operon. Okay, which we are in one part of this the DNA of the bacteria. Okay. So we have the regulator gene, okay, the gene that will regulate the expression of, for instance, for this particular system, the regulatory gene is actually the gene that will control, okay. It will regulate the control genes, and so it will regulate the structural genes to be expressed, okay. So the genes are responsible for microbial metabolism mechanism are organized in operons here. So this structure is called as operon. It must comprise of regulator gene, it must comprise of control genes. We have the control genes, basically it's comprised of promoter and so operator. And we have the structure gene that will be produced, that will be expressed when the metal is present. Okay, so the expression of the resistance gene is tightly regulated and induced by the presence of specific metal. So when the specific metal presence in the surrounding environment, so this expression of the gene will happen. And because of the specificity of this regulation, the promoters and the promoter and the regulatory genes from this resistant operon can be used to construct metal-specific biosensors, which is the promoter-reporter gene fusion. Okay, promoter-reporter gene fusion. Okay, so as I did mention to you earlier, once this structural gene has been replaced with the, for instance, the luciferase gene, okay, in table 1, or LUX gene, okay, this promoter change to we change the structural genes to luciferase, to LUC, okay, to GFP, or so on and so forth. Okay, when the regulatory gene sense the presence of metal, it will start to instruct the control genes, promoter and operator, to express the LUC and so luciferase, okay, to